Welcome back. So now let's look at another application of graph theory, a very different application in terms of predicting novel metabolic pathways using molecular graphs and what is known as subgraph mining. So what is the question? Can you predict novel metabolic pathways from reaction networks, from databases and so on? This is a very classic question. You see two molecules, two organic chemistry molecules, as you may remember from your school classes, and you have one molecule on the left hand side and another molecule on the right hand side. And this is a question you may have answered in school. You wouldn't have had these protein pictures there, but this is a question, right? So I have an A and I need to convert it to a B, and how do I do it? One step or multiple steps? How would you go about doing this? So typically in school this comes from your excellent memory of organic chemistry reactions. You say that, oh, I should do a Diels-Alder reaction or a Kolb-Schmidt reaction and so on, and convert the molecule on the one hand to the molecule on the other hand. Whereas today we can actually leverage the knowledge that is available in various biochemistry databases to do the same trick. So there are 10,000 plus reactions in various kinds of biochemistry databases. There are databases such as the KEGG and the BioCyc that are very popular, which essentially represent the repertoire of biochemical conversions that we know today that enzymes can catalyze. So there is an enzyme that can convert this molecule to some other molecule, and then there is another enzyme that can connect this molecule to this molecule perhaps. And a lot of metabolic engineering, there is this field of metabolic engineering which involves producing commercially important molecules using microbes and so on. This involves this notion of copy pasting or importing what we would call as non-native reactions. So these are reactions that don't exist in a particular species. You copy paste them, right, from species 1 into species 2 so that you can perform some new reactions and produce novel metabolites in species 2. This is called retrosynthesis because your question is centered around this molecule. How do I synthesize this molecule from either this molecule or any known starting molecule? I could just say glucose, or I could just say, hey, tell me how to produce this particular molecule in this organism. Right? I know the chemistry of this, biochemistry of this organism. I know it can produce a few thousand molecules, but I am not sure if it can produce this, or if it can produce this, what are the ways that you can possibly produce this. And if you look at this, typically a lot of the existing approaches involve the knowledge of these pathways. So I know that this molecule exists and this molecule exists, and you try to predict pathways. But if you want to predict novel pathways or new pathways, especially to molecules that, for example, may not exist in databases. So assuming your right hand side molecule does not exist in current databases, it becomes all the more interesting and challenging. So how do we go about this? Can we build a fully automated engine that predicts these pathways between these molecules, between a pair of molecules? Right? The next step is to generalize towards an unknown molecule on the left hand side, but let's at least start with a pair of molecules. You can possibly even pick it based on what is most chemically similar to the right hand side molecule in your existing metabolic space corresponding to your organism of interest. And can you do this relying only on structural information, meaning you do not have thermodynamic information, you do not have reaction mechanism information and so on, or what atom is getting transformed to what atom and things like that. Right, so this was our approach called reaction miner. It's a very interesting approach where we represent every molecule as a graph. So this is the departure from what we were talking so far, right? We were talking about every molecule being a node in a network and things like that. We talked about protein interaction networks or metabolic networks where every node in the network is a molecule. Now every atom in a molecule becomes a node. Originally we had every molecule in the organism being part of the network, right? A node in the network and two molecules that interact were connected. Now your molecule itself is the network. Dive deep into it. And every atom in this molecule is a node, and two atoms that are connected by a bond can be connected by an edge, and we also accounted for single bonds, double bonds, triple bonds, and bonds outside the plane into the plane, because optical activity is very central to biological molecules and so on. And we first performed a reactant product mapping. This is interesting because you have reactions of the form so here typically what happens is A gets converted to C, let's say, or it could be D also, and B gets converted to D, meaning A is sort of chemically similar to C and B typically is some sort of a helper molecule. It could be ATP or something like that, right? So this could be a reaction like glucose plus ATP gives glucose 6-phosphate plus ADP. Here glucose does not get converted to ADP, but fairly obviously glucose gets converted to glucose 6-phosphate. So if this is the conversion that's happening, you can learn what is the transformation that's happening. Essentially a phosphate bond, by the sound of it, it seems that a phosphate bond is being added to glucose and it is being lost from ATP. So can you capture this? Can you first identify what are the reaction pairs, and we perform this by using subgraph edit distance. So you take all pairs here that are possible here like AC, AD, BC, BD and so on, and then match those that have the shortest subgraph edit distance. So in this case, let's say if this was glucose and this was glucose 6-phosphate and this was ATP and this was ADP, the shortest distance would be between glucose and glucose 6-phosphate, or well, it could be between ATP and ADP as well. Very similar changes there. So these two molecules will start getting mapped together. So this helps us learn what is the transformation, essentially trying to answer this question. So there is this molecule with some transformation, it has gotten converted to this molecule. What is the transformation? How do you characterize the transformation? We characterize the transformation in terms of a reaction center, which is where the action happens, where some molecules, where the molecules atoms are replaced or removed and so on, and there is on the corresponding other molecule, on the product molecule, these atoms are added and so forth. So we identify the reaction center, which is the place of change, the reaction signature, which essentially tries to capture the neighbourhood of the atoms where this action is happening, because this is trying to roughly capture enzyme chemistry, because an enzyme goes and attacks a molecule that has a particular kind of chemistry. What kind of chemistry? Doesn't just look for a carbon atom and go towards it, right? It should be a carbon attached to an oxygen attached to something else. So we look at a brief neighbourhood of this atom in this molecule and use that to capture what could be the substrate specificity of the enzyme, meaning can this enzyme go and interact with this molecule. So this is a prediction, a model, but it works well in practice. And we identify reaction rules by summarizing the transformation in reaction signatures. So what is the change that happens and so on, and most importantly we take all these rules. So these rules essentially capture, I have some molecule on the left hand side, if it looks like this and has these kinds of bonds, then I can convert it to a molecule that looks like this and has these kinds of bonds. Essentially learning the transformation pattern between a pair of molecules. And we then embedded these reaction rules in another network again. So we are talking about multiple networks. We talked about molecular networks earlier. Today a molecule is a network, and from this molecule, using these molecule networks, right, networks as molecule as a network, and looking at the similarities and differences, the edits, the additions, the deletions and bonds and so on, we identify reaction signatures, reaction rules and so on. And we then take these, and I am not going into all the details, you should go and read the paper for it. I am just trying to give you a quick intro so as to pique your interest to go and read about these, because there are so many applications of network biology, it would be impossible to cover all of them in the course, but I am trying to give you a very diverse set of applications that will motivate you to A, understand how important biological problems can be tackled using network biology, and B, also see how network biology is so ubiquitous in its application, right? There are so many different places where simple network concepts can be readily applied to get very interesting insights. So you now create a reaction rule network where every node is a reaction rule and an edge connects reaction rules that can be successively applied. So you can learn and generalize using this network, and pathway prediction simply becomes pathfinding on this reaction rule network. So basically if you want to find how to make this molecule from this molecule, you find some signatures in this molecule and go back to the reaction network, look for those signatures, look at neighboring signatures and find a path to a node that contains signatures from this molecule, right? And there you go, you have a pathway, a set of reactions. You can fill in these question marks and solve the problem of retrosynthesizing this molecule from this molecule. We also include a couple of heuristics and so on to rank the most plausible pathways based on the similarity between the molecules and things like that. This really made us fish out a single important pathway from a very large set of biologically plausible pathways. Okay. And in this work, Arvind was the student who did most of this work, and Shayan Ranu, who was in the computer science department, was my collaborator on this. It was very interesting because we could, with Shayan's help, we could get really interesting data structures that we could use to speed up this search and so on. Otherwise, this was a problem that wouldn't even take off, and we eventually got very interesting results. So what did we find? We found we were able to recover naturally occurring pathways in many cases, even in central metabolism, which is essentially very confusing because central metabolism has a lot of similar looking molecules and carbohydrates, amino acids getting transformed to one another. And we also were able to get a realistic ranking of these pathways from a large pool of alternates. And we predicted the correct retrosynthetic pathway that has been found in literature and so on in many cases. The most important feature of our algorithm is that we used only structure information based on and no information on atom mapping or anything like that. We used only structural information and no information on atom mapping or hand curated rules that are typically present in other databases and therefore leveraged by algorithms. No thermodynamic information either. So this became a very interesting application of network biology in a very different way, right? So we used two different kind of networks here. We used this molecule as a graph, molecule as a network, and then we extracted reaction signatures and then put these reaction signatures into reaction signatures and reaction rules, and we put these reaction rules into a reaction rule network, essentially enabling rapid pathfinding for even very large very complex molecules and so on in very large networks, right? More than the molecule complexity. The molecule complexity also leads to lots of possibilities and so on, but we looked at very large databases. We also artificially expanded the database for future scenarios when our biochemical knowledge would improve, and we showed that our method was also highly scalable. So this is a very important application, and I hope I would have now convinced you that there are very different applications of network biology, and we will see more different applications in the next class.